I just meant actually now we're going. Yeah. That's all. This was
Okay, how's everybody doing? Good. How's the weekend? Okay, so what we're gonna be talking about today, we finished chapter 18. So the last chapter on the section of optics is about optical instruments. We talk about a little bit about microscopes. So the last instrument is basically the human eye. So between today and Wednesday, we're going to be talking about human eye. Today is just basically kind of the anatomy of the human eye. And Wednesday is going to be a short class about different kind of procedures that is being used to correct vision deficiencies. How many of you have taken anatomy? Oh, most of you. So what you're going to see here probably is a little bit repetition. You probably have seen this before. I'll want to show the human eye from the point of view of physics. So how do we analyze the human eye? How can we make a model of the human eye using thinness equation and using some of the systems that we have seen? And how can we correct some eye deficiency, side deficiency, like aperopia, myopia with the thinness equation? So that will be just this week, and then we're done pretty much with the whole section of optics. So yay. Okay, so this is a human eye. You probably have seen these schematics before. Obviously, the human eye is a very complex structure. There are elements that help them to give some mechanical stability. There are some elements that help the eye get movement and help direct the vision. The fact that we have two eyes helps us to have this stereoscopic vision and allows us to have depth of focus. There are other elements that are basically in charge of creating the image on the retina. So for this class, for this lecture, I'm only gonna care about the elements, about the optical elements, the elements that help to create an image from an object to the back of your retina. So I'm not gonna deal with how you create a stereoscopic vision or many of the elements that you have in the human eye. So the first component is the cornea, is the exterior part of your eye, is the one that you can actually touch. Don't try to touch it, Caleb. It's a very delicate structure. If you scratch it, it can affect your vision. And the main objective or the main function of the cornea is to capture light. Has the largest curvature of all the optical components. And basically, it's that first window that allows to capture light. So anything that scratches or clouds the cornea basically is going to affect the way you create the image. Many of the procedures that involves correcting vision, like LASIK, that's the one that's more popular, involves making mechanical modifications to the cornea. You basically burn it with a laser, you give it a different shape, and you can correct vision. So yeah, don't touch it, Caleb. One of the things that we care about these optical components is the curvature that defines the optical power. But the other one is the refractive index. I'm going to show the values of refractive index of some components. And you will see that most of them are around the 1.3 range, the 1.37, 1.33, 1.35, stuff like that. The refractive index of water, of the still water, is 1.33. With water and salt, it's around 1.34, 1.35, depending on the concentration of salt. So you can think that many of the liquids in our body, they are around the refractive index of water with salt. It's around 1.35, and you will see those numbers that are very, they repeat in different structures. Remember that when light goes from one medium to another, so when it goes from air to water or air to glass or something like that, we're going to have reflection, and we're going to have refraction. So is that bending on the light, and there's a bounce of light from that surface. The biggest change that we're going to have when light goes into the eye is precisely here in the cornea. That's why we have that cornea with a large curvature to kind of help that refraction and to help the light to focus on the back of the retina. If light refracts too much, especially if you have different colors going into your eye, what's going to happen is that you're going to have blue light focusing in a different position than red light. And then instead of having a clear image of an object, well, you have it's kind of like when you see a 3D movie without your glasses, that you can see kind of the red figure on one side and the blue figure kind of not matching. That's what happens in optical system. Like the figure doesn't match and you see kind of blurry. So many of the components that we have is basically what it's trying to do is making sure that every single color focus at the same point and to create this sharp image of the object. Okay, so for example, behind the cornea, we have the vitreous humor, the aqueous humor, and you can see, for example, that the refractive index is significantly lower. It doesn't seem like much, like going from 1.37 to 1.33, but that's a big change. This is more like a lubricant. It just prevents your cornea from scratching. It gives this ability of your iris and your lens to move freely. And it's this kind of this buffer zone that between the two components. Okay. We have the iris. The iris is the part of color that we see in our eye. It has different functions. First of all, we have a involuntary reflex on the iris. So depending on the amount of eye that is being shined into our eye, the cornea is going to contract, it's going to dilate. You will see in the movies, you will see every time you go to the doctor, the first thing that they do is that they check that reflex. They shine a flashlight in your eyeball, and they see that dilatation, and they see how fast it is. One of the first indications of having brain damage is the lack of that reflex. So it's one of the first things that you check if there's that if you have that contraction. When you contract and you expand the iris, it's not only that you limit the amount of light that goes into your eye, that of course, if you have low light conditions, you want to have more light so you can actually see what's going on. But also, remember when we were talking about diffraction, and we were talking about diffraction about an optical aperture, that there is a resolution, and the limit of that resolution is going to depend on the diameter of the aperture. So this, by controlling the size of your iris, you can control the resolution of objects that you see. So when you are focusing on something, when you are reading, or if you are trying to put a thread through a needle, or doing something that requires a lot of precision, the size of your iris is going to contract. It's going to change depending on what you're trying to do. So it's not only amount of light; it's also help us to focus. Drivers, when they're driving Formula One drivers, the iris kind of contracts and they have that tunnel vision. That's basically trying to focus or trying to have a very sharp image at the center vision, and you blurry everything else around. Now, one of the structures that is more interesting in the human eye is the lens. I just want to curious because that's what you do. You barely read. What the hell is that?
It's the game.
It's a game, and you have to pay. It has one of those microtransactions.
It has tax plays, but you. Oh yeah, wait.
One of the most complex structure is the lens. And to try to understand how amazing this structure is, I'm going to show you a photographic lens, just a regular lens. Oops, what happened to my? I used to have a photographic lens figure. I don't know what happened. Okay, I'll show you this. Okay, so this is just a regular camera lens. This is, I think, it's a zoom lens. So you can change a little bit of the of where you want to create that image, how close or far away you can focus that image. And all photographic lenses, they have different components. It's not just one single lens. So in this case, we have 1, 2, 3, 4, 5, 6 components, 6 groups, and each group has a couple of lenses. And each lens is in charge of something specific in the system. They are focusing the light. They're removing kind of aberrations. They are making sure that all the light is trying to capture as much light as possible. So every single element that we see here has a role. So when you remove one of them, your image is going to be all messed up. This is kind of a very common kind of lens. Usually they have maybe around 20, 24 individual lenses. If you see a football game or basketball game, on the sidelines you see the photographers, and the photographers have these huge lenses that the casing is white as opposed to the regular black. Those are telephotos of Canon. That specific is the specific brand. And those tend to be maybe 50 components, like different individual lenses. The lenses that are being used to fabricate transistors and chips, they may be like more complex, and they may have maybe 70 components. And the human lens, oh, there it is. What the hell? That's the one that I want to show. I don't know. The human lens is like an onion. So it has individual layers, and they are very much like the onions, like that kind of dimensions. And there are around 22,000 of these little layers. And each layer is going to have a different refractive index. So each layer is a lens. Not only is going to have a different refractive index, but the refractive index at the center is going to be different than the refractive index at the edges. We call that a gradient lens. It's flexible. It's an organic material. So we have a couple of muscles that surrounds the lens, and when those muscles contract, they bend the lens, so give it a higher curvature, so we can focus something that's close to us. And when the muscles are relaxed, this thing can extend, and then we can focus things on the back. So that effect that a telephoto does, that it moves the lens, the whole structure, make it bigger or shorter. The human lens created by bending our lens. So the refractive index of the lens goes from 1.406 to 1.386, roughly. So it's just amazing that it's just a very small structure, is a couple of centimeters or maybe less than 1 centimeter in thickness. And it has all these amazing optical power. It creates a very, very clear image of objects. Okay. Of course, because it's an organic material, it degrades with time. It can get cloudy with our cataracts. And obviously, when it gets cloudy, then your vision gets impaired. The muscles become weak. And as you go older, the lens is going to lose flexibility. So the ability to focus at different distances is going to be reduced. So you'll see that all people, we tend to need glasses to read. And it's basically because that muscles are not strong enough, or the lenses are not flexible enough to focus close to you. We have the vitreous humor, and it's just kind of like a liquid that is behind the lens. And this liquid, you can imagine that it's like a balloon full of water. That water can still play. Put that thing away. Just turn it over. There you go. I'm gonna keep an eye on that. Next time, I'm gonna put it in front. Think about the kind of like a balloon full of water. The water is going to give that balloon some mechanical stability. So it's going to prevent the balloon to change shapes or to get compressed or anything like that. It's just give it some mechanical stability. There's a lot of dead cells here inside. It's not water, technically what is there. There's a lot of organic material, so some parts get kind of harder than others. And all these gunk that we have on the back are going to create shadows. And those shadows, we don't see the specific shape of the things that create the shadow because they're behind the lens. So you need a lens to create an image. So there's no way we can focus that on our retina, but we can see the shadows. For most of the time, because the shadows are always there, we can ignore them. It's kind of like when you have white noise, or when you are in a class and you hear the professor and it kind of just fade away, and then you just ignore the professor and just like a noise in the back, kind of like that. And the noise is there, the shadows are there. You can see that if you see something very bright, like a white wall, sometimes you can see them. So if you stare and you focus, you can see these kind of floaters that they are somewhere in your vision, okay? So those are just dead cells that are just floating there. And because they're inside this liquid, it doesn't matter how you move your face, your head, they're always going to be moving with you. As you grow older, they are going to increase and they become very annoying. There's no way you can remove them. It's not like you can remove that liquid and put it back. The name in Latin means mucus, floating mucus, which I think is pretty cool. I don't know who come up with that name, but yeah, you can usually see them when you see something very bright, the white sky or a white screen. You can see those kind of floaters. And this is usually how we represent the eye when we want to make a physical model. Like we want to do the ray tracing that we did at the beginning, or we want to design an optical instrument. This is usually the system, the model of the eye that we use. We have the cornea, which is just a lens with a very, very high range of curvature and a very large diameter. So this thing will be my cornea. And then the lens, you can see that it's not the regular lenses that we use. We actually need to work with a thick lens, and also we need to work a thick lens with the cornea. So it's a thick lens, but it's not a symmetric lens. It's not like the biconvex lenses that we've been working. You have a range of curvature in the front, and then in the back you have a different range of curvature. Okay, this P prime, N prime, all this stuff is what we call principal nodes, or cardinal points. In any optical system, there are 5 cardinal points. We, in our class, we usually work with 2, which are the focal points. Those are cardinal points. And besides those 2, there are the 3, which are these guys over here. That's what they mean. And this is a ray tracing for the eye. Okay, and that's basically what we do. I have a file that basically is that thing. And whenever I need to do a design for an optical instrument, the only thing that I do is I just pull the file and put it in my design and see if it works. And that's pretty much it. The retina is the back of your eye. That's the electrical system of the eye. It's kind of weird, the retina. Like, I don't know who designed the eye, but I think it was a little bit drunk. So imagine that you have, when you design a camera or you design the CCD lenses or the CCD camera, you have your photoreceptors. They are receiving the light. This is the part that can be excited with the light, and then on the back you have all the electronics and all the wires that are gonna take you to your computer, or in this case, to the brain. That's what you will imagine that will happen, but in the human eye is different. The human eye, the photoreceptors are kind of flip. So the part that is sensitive to the light are on the back. So the light has to hit the back of the eye and then bounce back. And all the electronics, all the nerves are on the front. And you have all these nerves in the front, and then you need to bring them out somewhere. So, okay, I'm gonna remove one photodetector, and then I'm gonna bring my nerves and then connect it to the brain. So the problem is that now you create a blind spot. There's one part that you're not gonna receive light, you're not gonna have any photoreceptors, and that's basically this part over here. Not all eyes are like that. Not all human eyes are like that, but there are animals that they don't have that problem, that they can receive, they don't have blind spots, like octopus and nautilus and all these really weird animals. They have the correct eye, like what any decent people do. Mammals, many of us have that eye. So I don't know, someone in the evolutionary tree made a mistake. I don't know. We have 2 photoreceptors on the retina. We have cones and we have rods. And the cones are in charge of detecting color, and the rods are more sensitive to illumination and the intensity of the light and motion. And the distribution is a little bit different. It's pretty much unique to each one of us. The retina is kind of like a fingerprint. You can actually take pictures of the retina of a person, and it will be a way of identifying that person. It's that unique. This is how the retina looks like. I was going to bring my own picture of the retina. Don't look at me like that. I don't care if you're upset. You wait until the class is over to play. He's giving me stinky eye. So we have the optic nerve. So this is the blind spot. We have a second blind spot here on the fovea. And then you have all the nets of vascular systems. Have you noticed sometimes when you are making a really hard effort, like you carry something very big, or you stand up really quick and you're kind of dizzy, and then you can start seeing some flashes in front of your eye? Okay, so these vascular systems, these veins, are so thin that when blood goes through them, it's kind of like a train of red cells. So they only can fit one cell at a time. So they just go kind of like a train. And red cells absorbs pretty much all the light, so nothing goes through. But then from time to time, you have a white cell going through it. And the white cell lets all the light go through. They're kind of translucent. So when you see a white cell, you see a flash of light. And that's what you see, those flashes of light, when you make a big effort. So it's just kind of fun that you can see white cells going through your eye. Kind of cool. So you have the rods. You have a bunch of them, much more than cones. They are usually in charge of vision in the dark. So they don't perceive color. So if you are in a dark room, you don't see color. They're also in charge of motion. So you have them a lot in your peripheral vision. So if you see something or someone moving kind of almost behind you, you can to detect it. You're very sensitive to those kind of motion. The cones, on the other hand, they are also located. Most of them are located in the fovea. Most of them are located in the fovea, and they're in charge of color. We have 3 different kind of cones, and each cone is basically in charge of the detection of one wavelength. So we say that we have blue cones, green cones, red cones. Okay. Any one of you is colorblind? Anyone? Okay. There are people that are colorblind, so they are missing one of the photodetectors, or the photodetector is not as sensitive to that wavelength, and we should be. Usually, men have higher incidence of colorblindness. Joshua, I don't know which one is the one that you don't see. Is it red?
Predominantly, yeah.
Predominantly red. Usually, it's red, the one that is missing. And women, they have what is called tetrachromators. So there are a high, well, I don't know how high, but there's some women that have 4 photoreceptors instead of 3. And you can tell when someone is photoreceptors, when someone has 4 photoreceptors, by the way they describe colors. So the way they describe some elements. For example, if I see the sky as being blue. And maybe if it's a sunset, they will have different tones of red. But if I assume that it's just midday, clear sky, no clouds or anything, the sky is blue. But many women, well, not many women, but some women, they will see the sky with a tint of pink or with a tint of red. And when they see the skin of people, especially Caucasians, they see the skin not as pink or white, but with a tone of blue. And that's the way they describe it. It's really hard to measure it because all our screens and all our projectors are based on 3 colors. But that joke that men only see red, black, and red, blue, and green, and women see all the shades, it's not I mean, there's something in there. Like they are more sensitive, they are a higher sensitivity on the color, and it's because there's an additional photoreceptor. Now, each photoreceptor is going to be excited with a particular wavelength. Okay, and there are colors, for example, the yellow, that we don't have a photoreceptor for yellow. So if we want to perceive yellow, what happens is that the two photoreceptors, the green and the red, are going to be excited at the same time. And that combination of the two photoreceptors being excited at the same time gives the brain the impression that what is seen is yellow. And depending on which one is more intense compared to the other, then we're going to get closer to a red, orange, or more to a light yellow and stuff. There are some colors that they don't appear there. Brown, purple, they don't appear. And those are colors that our brain basically trick us to observe. There's nothing there, and because of that absence of color, it just basically creates a new one, and it gives you the impression of being purple. So let me show you a quick video.
We have this entire misunderstanding of what Darwin really meant when he said survival of the fittest.
Purple is a weird color. The formal name for purple is magenta. And the weird thing about magenta is that you won't ever see it in a rainbow. And the rainbow is supposed to be the full spectrum of colors. So why doesn't magenta appear in the rainbow? And the answer is to do with color mixing. I've always had a problem with color mixing because I know that you can't mix photons together. So you can't take a blue photon and a green photon and mix them together to get some other photon. That just doesn't happen. And yet you can mix paints together in art. Like, color mixing is definitely something you can do. So what's the answer? Well, actually, you can't mix colors together in physics, but you can do it in biology. It's to do with how your eyes work. For example, if I shine red light and green light into your eyes at the same time, if I cross these over, you will see yellow. So when you mix red and green together, you get yellow. And if you look at the spectrum, yellow is in between red and green. So maybe that's the rule for mixing colors together. You mix two colors together, you get the color in between on the color spectrum. And we can test that again. So I'll look at green and blue together. So if I mix green and blue together, I get cyan. And cyan is in between blue and green on the spectrum. So it's great. You mix two colors together, you get the color in between. But why? Why is that? Well, your eyes can't measure the wavelength of light directly. So it's not like a photon comes in and it's, 200 nanometers or whatever, and it detects that. Instead, you have these cone cells at the back of your eyes that are sensitive to different parts of the spectrum. So when red light comes into your eyes, there's a set of cones that fire and tell your brain you're looking at something red. So we'd call those the red cones. There's another set of cones that are more sensitive to green. So when there's green light going into your eyes, they fire and they send a message to your brain. And there's blue cones as well. So you've got red cones, green cones, and blue cones. So what about yellow? What about when you're looking at yellow light like that? Well, in that situation, you don't have a yellow cone. So what do? Well, yellow is quite close to red, so your red cone fires a bit. And yellow is quite close to green as well, so your green cone fires a bit. So your brain is getting a message from your red cone and your green cone at the same time. And it's deciding, okay, well, I must be looking at something in between those two colors then. And that's brilliant because your brain is perceiving something about the world that it isn't able to measure directly. It isn't directly sensitive to yellow light. It does mean that you can be tricked. And so if I make red light and green light go into your eyes, but no yellow light, you will see yellow anyway. So I've got a red torch and a green torch, and there's no yellow light here. But when I combine them, you will see yellow anyway. And TVs do this all the time. So if you look up closer at TV, you'll see the individual pixels, and there are red pixels, green pixels, and blue pixels. Those are the only colors being produced by your TV. And yet they can produce all the other colors with this trick of color mixing. So what about purple? What about magenta? Well, what should your brain do if your red cone fires at one end of the spectrum and your blue cone fires at the other end of the spectrum, but your green cone doesn't fire? Does it do the same trick? Does it think, I must be looking at a color in between red and blue? Well, the color in between red and blue is green, and you're definitely not looking at something green because your green cone isn't firing. So in that situation, your brain invents a color. It makes up a color, and that color is magenta. And I can show you that. So with my red and blue torches, so when they combine together, there you see magenta. Absolutely beautiful. And that's why you don't see magenta in the spectrum. You don't see magenta in the rainbow because it doesn't have a wavelength. It's just the absence of green, if you like. Just to show you the full palette of colors that you can see on a TV screen. So you get red and blue mixed together makes magenta. Green and red makes yellow. Green and blue makes cyan. When you mix them all together, you get white. So when your red cone, your green cone, and your blue cone are firing together, you get white light.
It is different how you see the color, depending if you are projecting the color through a screen. Like, for example, what you're seeing right now are basically these pixels on the projector that basically they make the red colors. When you see your phone or whatever, it's like these red pixels on your screen. The LEDs are making that red color. But when you see the fire extinguisher that is red, it's the opposite. You have white light illuminating that thing, and all the colors are going to be absorbed except for red. So when you see something, paper, an object, the colors, we say that is a subtractive procedure. All wavelengths are going to be subtracted except for the one that you want to see. When you are seeing through a screen, you are just illuminating the one's colors that you see. It's kind of weird. So different animals will have different photoreceptors. And also what is also kind of interesting with color is that the creation of color, or what we perceive as color, is not only about the physics and what cones are being fired or not, but it's also about culture, and it's also about language. If you see the evolution of languages, the colors that they are first described as, for example, are reds and greens. Then you have yellows, browns, whites, and then at the end of the evolution of the color, you have blues. In ancient Greek or the traditional Greek, they don't have a word for blue. So when you see translations, or when you people that can read the Greek philosophers and they read a language, they don't describe the sky or the ocean as blue. They usually do it with reddish or purplish or something different. They don't have the word for blue. It's blue is very unusual in nature. In animals, plants, it's not that common as other colors. So usually it takes a lot to long for the language to develop. And there's a lot of people that they describe that. Okay, how can you describe a color if you don't have a word for the color? How can you describe red to someone that cannot see red?
So in that case, with like the woman having 4 photoreceptors and seeing the sky as like blue and pink, would that just be blue to them, or would that be blue and pink to them? Does that make sense?
For the people that they are tetrachromatic, they see a different color, but they don't know there's no word for that color. So the best way that they can describe it is that it's bluish, pinkish. For us, the people that only have 3 photoreceptors, for us will look like blue because we don't have that extra photoreceptors to perceive that changes. Makes sense?
Yeah, is there like a specific color that fourth one would be? Or does it like change like, cause you said that we all have like, red, green and blue, I think you said. So does that always have to be like red for every person who has a fourth photoreceptor, or can it be blue or green?
Oh, I don't know. Oh, that's a good one. I don't know. I'll look around. Colors, they have two photoreceptors. Colors. Dogs have two photoreceptors, usually green and blue. So they don't see any that has to do with yellow, oranges, anything like that. Humans, we have three photoreceptors, and we can see the combinations of all of them. Different animals, they have additional photoreceptors that they extend beyond the visible range. So many insects, especially those that are use some kind of pollination, or they have some nectar, or they look for nectar on flowers, they see in the ultraviolet, because a plant that has pollen and that has nectar looks different on the ultraviolet than it's not. Reptiles, they can see on the infrared, so they can identify prey and they can identify hotspots. So they are more sensitive to the ultraviolet. Animals that hunt at night, they all can also see other kind of colors. And I show you the video for the mantis shrimp, so I'm not gonna do it now, but that thing can see like 16 receptive colors. Okay, the macula lutea, this little spot over here, this is what most of the cones exist. That's where we have them. It's a very sensitive area. The cones are very, very sensitive. They can be excited really, really easy. And one of the things with photoreceptors is like when you hear a really loud sound and then you have that ringing on your ear, and they can stand. Something like that can happen to the eye. You shine it really high, really high intensity, and you don't protect the eye. Your cones kind of stay excited, and they start creating kind of fake artifacts on your vision. So if you see that image seems that it's moving. It's moving because of two things. One is because the colors are very bright and your eyes are getting overstimulated, but also because your eye is trying to move away the image from the macula, from the fovea, so it doesn't damage your cones. And that micro movement creates that sensation of motion. In this one, you can see it a little bit better. If you focus on the center one, you can see that kind of jumps around. That's your eye moving, which is kind of cool. I'm just gonna leave it there until you guys get this. Okay. Most of the color, most of the things that we perceive is that it is on the macula lutea. So most of the cones are there. Cones are not sensitive to low light intensity. So when it's dark, we have a second blind spot over here because we have very few rods that they are the ones that are being activated in light vision and night vision. Okay, makes sense? Okay, so within eyes sight defects, there are different ones that we can have. We can have astigmatism, presbyopia, I don't know. There are myopia, hyperopia, two of the more common ones. So many of them are based because the lens is too stiff and we lose some focal power and we cannot focus the right way. Some other ones is because the shape of the eye is not perfect. And then the lens doesn't create the image at the retina. So for example, myopia is, for example, if you see something very close, you can see it pretty well, but if something is far away, you don't see it. It looks blurry. So on a healthy eye, when you can see things that are far away. You see them clearly. You may not be able to see all the details, but you should see them. So, for example, in my case, if I remove my glasses, I don't see you. Well, I do see you guys, but you're all blurry. Like, I know that you have a nose and two eyes. That's it. I may distinguish you because of the hair, but that's it. I don't know if you're smiling, have your eyes closed. I don't know. So my far vision, instead of being at infinity, is somewhere closer. Okay, so I'm gonna try to find where is that far vision. Okay, so Mason, don't freak out. I wanna walk towards you. Okay, and I'm gonna walk until I can see you. Okay, so he's blurry. He's blinking. I can see that. Still blurry. Yeah, he's wearing the creepy. Still blurry. There he is. Okay, so things that can be an infinity for me is arm's length. Okay, so my infinite vision, something that is an infinity, I cannot see it. So my glasses, what they're trying to do is an object that is an infinity, they're gonna create an image that is at arm's length. And that's what I'm gonna see. Okay? So that's what the glasses are gonna try to do. So I'm just gonna use the thin lens equation. So my object is gonna be an infinity, and I'm gonna create an image that is gonna be, I mean, this is, it's not 2 meters, this is like a meter for me. So let's say that's 2.5 meters. So that's where my eyes can focus and they can create a clear image. And from here, I'm gonna find the inverse of the focal length. So doing this, I get negative 0.4 meters inverse. Okay, this meter inverse is what we know as a diopter. Okay, a diopter is a weird unit. It's a French, and they developed it before the French Revolution. And we just kept it. Don't ask me why. We just kept it. So when you go to your doctor and they give you your prescription of your glasses, and they tell you, oh, you have a 0.5 diopter, what it means is that the inverse of the focal length in meters is 0.5. That's what it means. Okay, but that's what, when you want to correct myopia, that's what you do. An object that is an infinity, you want to bring it to the far side of your patient. In my case, it's to 80 centimeters. For these particular examples, at 2.5. Aperopia is the opposite. Something that should be at a comfortable length of 25 centimeters. It looks blurry, and you need to move it away so you can see it. And if you have your parents or grandparents, you can see that when they try to read something, they always kind of moving their hands, and there's like, oh crap, my arms are not long enough. That's basically what I'm trying to do. We usually can see things very close. You can grab a book or your phone and you can put it at a comfortable distance, but your eyes cannot focus anymore. So you need to move the image far away until it focuses. So what the glasses are going to try to do is that an object that is close to create an image that's far away so you can focus. So you want to place your object, you want to place your book, whatever you are reading, you're going to put it at 0.25 centimeters. That's the near side, that's the normal near side. And you're going to create an image farther away where your eyes can actually focus. And that's going to give you your prescription. Okay. Notice that in both cases, the image that you are creating is a virtual image. The image that you're creating is in front of you. It's not behind your lenses. So we have a virtual image here, and we have a virtual image there too. And it's always kind of weird to understand, but yeah, your glasses are creating virtual images. So every time we see something through glasses, we are looking at virtual images. Okay. Questions? Make sense? Cool. Okay. Have a good day. See you guys on Wednesday. You better win that tournament or whatever you're doing.
All right.
Have a good day, everybody.